Good morning to everybody. So, talking about DNA barcoding. DNA barcoding is a molecular technique for the identification of living beings. The idea is quite simple. When you go to the supermarket and you are buying some items, you can identify the item by its barcode. So you know that it is exactly that part of things of the supermarket. So the analogy is that in an organism, like in living beings, you have DNA, and a small part of DNA can be used, and of course, in analogy, like a barcode to identify the organism. So this is the idea, which is not really new for biology, because we were already using this approach since decades. But the new things about the project, the barcoding, is that now it became let me say, a very ambitious project, because the idea of the DNA barcoding project, so coordinated by the international consortium, is to give to all the biodiversity, the world biodiversity, a tag, what we call a tag, so a molecular identification. So to combine the biodiversity to molecular variations. So the idea is quite simple, and it's a very difficult then to collect all the samples and to do this thing to all the organisms. But since a few years, 5 or 6 years, we have organized this consortium, and the basic new things of the consortium of the idea is that we can use DNA as a barcode. So of course we have not a real barcode within the DNA. It's just an analogy, because sometimes people are asking me if there is the real barcode inside the DNA. So the idea is to try to identify organisms not only, as usual we have done in biology, from all the form, the morphology, but also to work on fragments, on part of the organism, not completely. So this is the first innovation of the idea. So the possibility to give a name to something that is not only a part of what is like here, the one that you are eating now, but also to different stages of the organism. Because you can work also on juveniles, which are maybe different from the adults, so we are not able to identify easily. Or you can also work on small derivates from these organisms. Or other things, you can also have matrices, complex matrices, in which you have mixed different organisms, and so you can try to identify these organisms using this kind of approach. So the other important thing of this approach is the standardization. So for the first time, we are trying to put together all the biologies, because it was very usual for biologists to work in a separate areas, because people working on plants and people working on different animals were not using the same way to identify organisms. In DNA barcoding, the idea is to use what we call standardized part of the DNA. So the idea is that you have not to know what there is inside your food that you use now. There are different dishes in this moment in your food. So it's not necessary to know what you have inside. But with this technique, you can identify what there is inside. So the possibility to work also on complex matrices. So in our lab, we are working on basically to identify almost all the big kingdoms. So we are working on plants, we are working on animals, we are working on bacteria. And we are applying this approach to identify these different organisms. Of course, now I want to introduce you into one of our research, which is linked to seafood, what you are eating in this moment. And so, in seafood, we think that is very interesting to work with this approach, because very often you have fish in these conditions, so already managed. So in the slices or in part, in any case, in a condition where you cannot use morphology, in any case, to identify these things. If you are a consumer, you have to trust people what they are selling you. They are saying that, okay, this is, let me say, one kind of fish, that is another. You have to trust the people that are selling you. And the point is quite interesting, because in seafood you have many species that can be eaten. Because it's not a problem only when you are eating, let me say, beef, you know that there are few species that you can eat. But if you are eating fish, or seafood in general, not only fish, you have hundreds of species that can be sold in easy way. So the idea is that even if they are selling you something that is not what they are saying on the label, with using DNA barcoding there is no problem, because the juveniles, the part of the organism, all of them have inside this molecule, DNA. So it's not a matter of what we are working on. The point is that everybody has the same barcode, so we can identify all the things together. And so it's quite easy to work with using this approach. So what we have done in our lab and about seafood, we have worked on the Italian fish market, and in particular on Milan fish market, which is one of the biggest in Italy. We have not seen on Milan, but we have an important fish market. And so we have identified some key species, let me say flatfishes or perches, another important fish for Italy, or another fish which is called palombo, which is very common in some of our recipes. And we are the biggest consumers in the world of these fish. And what we have found is that, for instance, this fish for palombo, is that in Italian Milano fish market, around 90% of the label that you are going to buy when you go to the fish market is not the correct fish. It is another 16 fish that you are buying with the same name. Of course, it is a point of, as a matter of economical problem, because of course many of these fish have not a real value from the economical point of view, so they have to have basically no quotation for being sold. But on the other point, you have also a problem because sometimes we found also endangered species. So that you are eating an endangered species without knowing it. And then there is the possible problem with the allergens, because you think you eat just one fish, but you are eating 16 different species, sometimes very far from the biological point of view. So it's a real big trouble. So what we have seen with our method is that consumers can have an opportunity to know what they are eating, because now technology makes these things very cheap. They can do, using lab like ours, they can have an idea of what they are eating in a few days with a reasonable cost. So it's a good technique, and a technique that can allow you to know better what is around us, something that we always want to know, but that we are far to know, to be really honest. Okay.
Could you use it to identify even pieces of whale meat, just like this one, in say, Japanese restaurant or something, a sushi restaurant? Thank you.
Yes, it took us some time to set up all of this for this presentation, but I see the value for this.
So, yeah, you are completely correct. The piece of DNA should be something that is inside all the organisms. So, in this moment, we are using a piece of DNA that, in particular for animals, that is inside the mitochondria. So, this part of the cell. So, every cell, the organism has mitochondria. So, we are using a part of the genome of mitochondria, which is common in animal and plants. So, we can work on all of these. The only trouble is that it's working quite well for animals, not so well for plants. So for plants we are not able to identify all the species. So for plants we have to use a combination. It's not enough. You can say that you are in a certain part of the plants, but you cannot say the right plant. So you can use from the beginning this molecule by what you are seeing, then you can use another molecule to be a little bit more. And from the second question, whale, of course, yes, because it's the idea. It's easier when you have just on the sushi. It's very easy because usually you have just one species, so it's not a real problem. When you have more species together, it becomes a bit tricky. So the only problem is that when you have a complex matrix, but when you have a simple matrix, there is almost no problem, in particular for animals.